All right, moving on in our different microflora, we come to bacteria. Bacteria are their own group of organisms, and if you take microbiology with Laura, you'll learn a whole bunch about them. Some are aerobic, some are anaerobic. They produce asexually, they come in different shapes, so they have them in little cocci or cossi, little balls. They're rod-shaped, and sometimes you even get spiral-shaped bacteria. Some of them have flagella that help them move around. Some have pili, little tiny hair-like projections on their surface. When we look at them in the lab, this is what we're seeing, are these little purple and pink dots and lines. We're not getting to see a lot of those other projections. So those pink and purple dots and lines are because of gram-positive and negative assessment. This is one way we can distinguish bacteria apart. Some bacteria have a really thick cell wall with peptidoglycan layers, and that thick cell wall will hold a crystal violet stain. So we'll take our bacteria sample and we'll wash crystal violet over it, and it'll turn purple. And then we'll wash it away. And if it's gram-positive, that violet stain won't wash away. And you'll get these purple bacterial cells when you look under a microscope. But other cells are lacking that cell wall. Instead, they have a really thick cell membrane. So that means the violet gets washed away, and then when we add the red counterstain of usually red saffron, it gets trapped in the cell wall. So you get this pink or reddish gram-negative. So if it is gram-positive, the violet stain stays in the cell wall and the red stain doesn't stick when you add it. But if it is lacking that cell wall, the violet stain washes away, and when you add that red stain, it sticks. So gram-positive is purple, gram-negative is red. And I try to remember positive purple. But it's a great way to, when you're trying to distinguish what bacteria you have, here's a nice grouping. Is it gram-positive? Is it gram-negative? You've just eliminated half of the bacteria. Good place to start when you're trying to ID bacteria. So bacteria have different ways of feeding. We typically group them into autotrophs and heterotrophs. All bacteria are trying to get carbon in some way. Autotrophs are getting that carbon from the atmosphere, just like trees. So they're getting their energy from the sun, or sometimes they can get it right from inorganic compounds. So they can get that energy by breaking apart things like iron. And they make their own carbon by fixing it from the CO2 in the atmosphere. Heterotrophs get their carbon by decaying things. So these are bacteria that break down other organic material to get that carbon to use to make all the things they need. So probably the most famous bacteria in the world is E. coli. So this is what it looks like. It's a rod-shaped, gram-negative, so red-stained. It can grow in aerobic and anaerobic environments, and we find it in the soil. It grows best at body temperature, so when you get it in your body, it proliferates and can make you sick. But you poop it out, and your poop is in the soil. Well, maybe not your poop specifically, but animal poop is part of what makes up soil. So E. coli is a common soil bacteria. Another common soil bacteria is Clostridium botulinum. This is where botulism comes from. These are very common soil bacteria. They're anaerobic, so they're pretty micro amounts in the soil, but when they get into an anaerobic environment, they proliferate. So if we put them if they're in the soil and you're growing your carrots in the soil, and then you take your carrots out and you wash them off and you put them in a can and you haven't disinfected everything properly, then that botulinum bacteria can grow in that can, and all of a sudden you've got enough to kill a human. This is possibly, I think it's the most toxic natural thing that we know of. The next bacteria I'm going to talk about is B. subtilis. This is a common bacteria used in research because it's an important soil decomposer. It's also part of a healthy gut flora, and it can create this really tough endospore in order to survive rough conditions. So it's one that you might see on your Petri plates. It's also one that you might have heard of if you read any published papers on bacteria. And then the last one I want to talk about is the rhizobium that is found in the legume family. This might be the most famous nitrogen-fixing bacteria of all time. It creates a symbiotic relationship in these little root nodules of plants, specifically legume plants. So this is everything from like peas and alfalfa to acacia trees. Lupins are part of the legume family. All of the beans and lentils you've ever eaten are part of the legume family. It's a pretty large family. And they're all really good nitrogen fixers because they have this particular bacteria that grows symbiotically. So farmers learned about this years ago, that if you planted your field with peas or beans or alfalfa, your soil health would be better. You don't want to just be growing corn constantly. You want to cycle through and change out the plants because you want to allow those different nutrients to be cycled through your soil.